time for grade 12s, Learn Extra Live, Life Science. We have Aslan here with us today, and it's going to be amazing. I hope that you've been watching grade 10s and the 11 show just to, you know, brush up on everything that you know. If you have missed out, this is the most important site that you need to know for the year, especially you guys, the matrics of 2013. And it is www.learnextra.co.za/live. And what that's going to take you to, it's going to take you to a schedule, it's going to take you to the notes, which you can download for this very session and work through. And in a few days' time, the video will be uploaded there too onto YouTube. So this is the link that you need to be referring back to throughout the year. This show is also proudly sponsored by Macmillan. And here's the awesome Life Sciences Solution for All textbook, which all the content comes from and we'll be going through. Don't forget to post all of your questions onto www.facebook.com/learnextra. And you can also chat to me, Indy, on Twitter @learnextra. We are here to help you with any questions you may have. So now let's get straight to the show. Aslam, take it away.
Thanks, Indy. I see you're having moss and tea.
Moss and tea. This is from Llewellyn. Llewellyn's given me the moss from the grade 11 show. I'm going to put it into a little terrarium and I'm going to love it forever.
Okay, as long as you put it in the terrarium and not in your mouth.
No, I won't.
Welcome, Mindsetters. We are busy. We finished last week. We ended on the note on DNA replication and a little bit about DNA. We're now moving on and we're focusing on RNA and protein synthesis today. But before we go there, we need to just go a little forward in terms of let's just look at what we're looking for the whole day first, for the whole program today. First of all, we're going one step backwards, back to DNA, to cover the section on DNA profiling. And we want to look at DNA profiling, its use in forensics. What are the costs? What are ethical considerations we need to do there? And if we make mistakes, what are the consequences of that? Secondly, then we're starting with the RNA and protein synthesis. The first thing we look at is the location and function of RNA. That's a messenger RNA and transfer RNA. The structure of RNA, that's the next thing we look at. And obviously, we look at the different nitrogenous bases. We also want to look at, because we studied DNA and now we're going to study RNA, we also want to know the differences between DNA and RNA. We also then move on to say, because we're learning about RNA, more importantly, to see how RNA is involved in this process called protein synthesis. We want to know where does protein synthesis take place, how it takes place, and why does it take place in the first place. There's a lot of places in that statement that I made. Gene mutations thereafter, and then after DNA sequences. Gene mutations is what goes wrong with the genes, and sequences, how do we use the sequences in different DNAs of different organisms to show the relationship between them. So this last part is related to evolution. And you have covered that in grade 10 and 11. You've covered a little bit of evolution in grade 10 and you followed up in grade 11. You learned about phylogenetic trees and the relationship between organisms. So this is linked to that. This is another thing I'd like to stress while I'm talking about that. Don't look at life science and all the topics that you do in isolation. Every topic is leading towards something else. We're talking about DNA and protein synthesis, etc. This is also involved in evolution. It has a link there. It has a link in reproduction. It has a link in meiosis. It has a link in genetics. So can you see that one topic has lots of links across all the topics that you do. So don't look at these topics as islands. The main thing is that each of these topics are interlinked. And life sciences questions normally want to see whether you understand the link between all of these things. Last week we spoke about the structure of DNA, and we said that the sequence of the nitrogenous bases determine the structure of the DNA. We also said that the DNA that's in your body, the structure of the DNA, that means the fact that it has a phosphate, the fact that it has a deoxyribose sugar, the fact that it has nitrogenous bases, this structure of DNA is common in your body, a plant, that moss that's in this table, the flower, the rose tree in your garden, the cat that's walking around, jumping on you, all these organisms, the structure of DNA is the same in all of these organisms. The sequence of the nitrogenous bases are different, and that brings about the difference in the species that we'll talk about later. Now, when we're talking about DNA profiling, this was first done in 1987. 1987. Oops. Where was it done? It was done in the United Kingdom, in a little town called Leicester. I can't say very little because it's quite a huge town, Leicester, anyway. My daughter stays in Leicester as well. This professor, Sir Alec Jeffreys, he was knighted because of his discovery. In 1987, he discovered how we can use the DNA to identify criminals. And it was used the first time in 1988 in a criminal case. Later, there were famous cases where it was used. The most famous in our time being that of O.J. Simpson. Let more a little bit about that later. First of all, we must know that every human being, Indy sitting there, I'm sitting here, Belinda sitting in the background, James sitting in the background, all of us have unique DNA. Unless, of course, we are identical twins. Identical twins will have the same DNA. Why, you ask? Because they came from the same sperm and the same egg. So the one egg was fertilized, and this egg splits as it develops, and that then forms two babies in the end. That is why they are identical. You would then also ask, but every time I see twins, not all of them are looking the same. Yes, those may be fraternal twins. Those twins arise from a separate sperm and a separate egg. That means two sperm and two eggs. If they are triplets, it will be 3 sperms and 3 eggs, and so on and so on. So each individual has a unique DNA, and because of this, Sir Alec Jeffreys then started the process to say that we can use this to identify people. Now, how often have you gone to a store to buy some item or the other? Most of us are going in daily to stores. And most of the stores that we go in, they're not seeing, right, this is a bar one, and they tell you what's the price. They have a scanner. The scanner scans the barcode on that item, and immediately on the screen, if it's a bar one, it will show bar one. If it's an ice cream, whatever ice cream you're buying, or if it's Tipex or whatever you're buying, it will show that item. So each item in that particular store has a unique barcode. And that barcode, ladies and gentlemen, or boys and girls, whatever you want to call yourself, will be the DNA fingerprint of every individual.
Aslam, what happens if, this is from Nsako, she says, if we clone a human being, will they still have the same DNA?
Great question, Nsako. Nsako, that's a great question. What a question at the right time. She wants to know, if we clone an organism, will that organism have the same DNA? Yes, of course. What is a clone? A clone is an exact copy of an organism. And how do we clone an organism? We use that organism's DNA and the cells to make more cells. And therefore, any individual coming as a clone will have the identical DNA.
And that's why cloning is such a controversial thing, because we can't have 2 Indianas, because there's only one of me.
And one of me.
And one of you.
Okay, there we go. But it would be useful to having 2 Indianas.
It would be
Think
so
about it.
useful. It
You
would
could
be
be
so
taking a break
useful.
now.
Exactly, I know.
There we go. There's lots of pluses and there's lots of negative side of that, and we're going to talk about that today. So DNA profiling is then the technique that we use to get the DNA fingerprinting of or fingerprint of a person. You want to know, where do we find this DNA? We find this DNA in any body tissue. For example, saliva, blood, skin, semen, hair. If you look at this crime scene, and there you see on this victim there's some blood, that blood can be used to get DNA sample from there. Here's another one here with a knife and some blood, also the same situation. Or if in a case of rape, or if there's a murder where there was a struggle, somebody was being strangled, and the victim manages to scratch the perpetrator, then we can get some skin under the nails. From there too we can get that. And I love this last one here. You wouldn't be able to read very clearly. The lady is saying all the hair samples we found at the crime scene belong to you, detective. She's talking to the detective. And he says, yeah, actually I have got this hair loss problem. So his hair was found at the crime scene. This is how we do the DNA fingerprinting, guys. Very simple in one diagram. Again, something I like to get you used to. Don't just look at chunks of information, whole paragraphs and pages and pages of text and try to remember them. Try to look at the illustrations, because pictures say much more than words. And there we have it. What happens is when we get the DNA sample there, it is removed from the sample or from the victim or from the crime scene or wherever else we need to take it out. And this DNA is cut into several pieces using special enzymes called restriction enzymes. So they are chemical scissors, if you want to call it that. They cut the enzyme into the DNA into several pieces. This then is put into a structure there, a well with some gel inside, some liquid inside. And you'll notice there's a minus sign and a positive sign, a negative sign and a positive sign. That is the negative end of this unit, and that's the positive end of the unit. The DNA is loaded on the negative side. And we then pass current through this equipment, and the DNA fragments move towards the positively charged cathode. They move on towards the other side. Why? Because DNA is negatively charged. So unlike attracts, so they move in that direction. The smaller DNA segments move faster than the larger DNA segments, and therefore they move further away. They get to the end towards the positive pole faster. And in that way, as you can see, a pattern has been formed here. When it forms this pattern, they used some form of filter paper, and this the pattern is then copied onto the filter paper. From there, it goes onto an X-ray or something like that. And there we have what a DNA fingerprint would look like. You can see the different dark bands there. Those are different bands of DNA that is found there. Now, you can see already from this diagram that it's very similar to a barcode. Or should I say rather, the barcode is actually very similar to our DNA, because the DNA was there first. And people found that if individuals have unique codes and they can be identified by that, they then came up with the idea, we can use this for products. And look at what it has done to the product market now. A trader doesn't have to worry about remembering every price. He simply scans, and there comes the price on the board. And that, people, is the story of DNA, and it's the DNA fingerprinting. However, as Indy brought out earlier from the question on cloning, there are arguments for and there are arguments against DNA fingerprinting. First of all, DNA fingerprinting is a very expensive process. So therefore, especially in South Africa at the moment, we are not relying too much on DNA profiling or fingerprinting. You'll notice that only in the high-profile cases do we turn to this type of investigation. The rest, we're relying on fingerprints and pure detective work. But in other countries, it's already at a high stage. So that's one problem. It's very expensive. Secondly, we said earlier, if mistakes are made, the consequences are great. If we convict somebody because of a mistake a detective made, that person stays in jail for a long period of time unnecessarily. So those are some of the disadvantages. Advantages would be we can solve crime, we can solve medical issues, we can find out who's the father or siblings, etc. So those are the issues around DNA fingerprinting. Guys, I think with that, I've told you too much already. Over to Indy.
Yeah, we'll let them have a little think about that. It's like watching all those crime show those crime show channels. I love it. So if you are just as interested as I am, guys, make sure you keep stuck to your seats. Get yourself a glass of water, maybe a cup of tea. Just have a little bit of a relax, and we'll see you now after the break. Hey guys, and welcome back to Learn Extra Live. Matrix, I hope that you are paying attention and you are enjoying this lesson as much as I am. I'm loving, it's like a little bit of a debate. Cabello has said on the page, Indy, trust me, you don't want to clone. You know what? I am going to trust you, Cabello, and I'm going to make sure that there's no cloning of me anytime soon. I think what I'm going to do is I'm going to have a look on the page. What I need to do is please post pictures or questions on the page if there's anything that you need help with, and please make sure that it's relevant so we can make it go nice and fast. I think, Assam, let's carry on.
Let's go on. Guys, we've covered DNA. Now we're moving on with the core of today's lesson, and that is the RNA. So RNA, ribonucleic acid. Remember, we studied DNA, deoxyribonucleic acid. All right? We talk about the differences as we go along. All right. RNA, first of all, is single-stranded. The word polynucleotide we discussed last week, remember? Many nucleotides, so it's a long chain. However, RNA is not as long as DNA. So keep that in mind, because I'm going to ask you a question a bit later. So first of all, it is single-stranded. Secondly, it is long, but not as long as DNA. Second, further on, the nucleotides of RNA differ slightly from those of DNA. An RNA nucleotide consists of a ribose sugar. In DNA, we had deoxyribose sugar. A phosphate. In DNA, we had not D-phosphate, we had a phosphate. A phosphate we have in DNA, we have a phosphate in RNA as well. Always look at similarities and differences, because they can ask you for provide the similarities and differences, or provide the similarities only or the differences only. Okay, and they have the 4 bases. In DNA we had 4 bases, here too we have 4 bases. We have either uracil, cytosine, guanine, or adenine. Now which of these, guys, is different from last week? You're thinking about it? Good. You must have found out now that it is uracil. In DNA, there was no uracil. And uracil in DNA was replaced by thymine. I'm writing it here clearly that it's in DNA, so you do not get confused. That's in DNA. Remember, last week we spoke about the base pairing at cute girl. At adenine always combines with thymine, and cytosine always combines with guanine. So here we do not have thymine, so we have uracil. Keep that in your mind as well. If we look at the structure, there we have it. Again, a picture speaks much more than the words. Okay? There we have the phosphate. Look at the skeleton, the backbone, compared to last week's one. It's the same. Phosphate attached to the sugar attached to the nitrogenous base, and phosphate, sugar, nitrogenous base, and so on we go down. Difference being that there's no other strand on this side, so it's a single strand, number 1. The other difference being uracil there instead of thymine. And as you see, that's the structure of RNA. Now, the other difference is that RNA has several types. But again, somebody may argue, because of the discoveries we've made recently, there are different types of DNA as well. Remember from last week, we had nuclear DNA, mitochondrial DNA, and chloroplast DNA. So there are different types of DNA as well. However, there are specific different types of RNA, and they are, firstly, messenger RNA. Secondly, ribosomal RNA. Notice the abbreviations used. These are standard abbreviations. You can use it in your writing of your answers. mRNA can be used for messenger RNA. rRNA can be used for ribosomal RNA. And tRNA can be used for transfer RNA. Now let's look at each one. And guys, if you listen carefully here, and if you understand where do we find messenger RNA, ribosomal RNA, and transfer RNA, And what each one of them does, that explains protein synthesis. It's as simple as that. If you can understand where we find them and what they do, what's their function, what they do, what are they there for, that, the three combined, gives you the whole of protein synthesis. You don't have to worry about a thing if you learn this properly. So let's listen very carefully now. Messenger RNA, first of all, carries information about the amino acid sequence from the DNA. In other words, messenger RNA copies the code from the DNA. That's the first thing. And we're not going to go into detail now because we'll go into that detail later. Ribosomal RNA, they don't have a real shape as such, but they form in the ribosomes, and the ribosomes are formed in the cytoplasm of the cell that make proteins. In other words, ribosomes are the site, or is the site of protein synthesis. Last one, transfer RNA, small molecule, clover leaf shape. It picks up amino acids. Listen to this. It picks up amino acids. From where? From the cytoplasm. And what does it do with them? It brings them to the ribosomes where they are joined together to form a protein. So tRNA is basically like a taxi. Its job, go to the taxi rank, pick up the amino acids, bring them to the marketplace. In this case, the marketplace being the ribosome, because that is where the transfer RNA need to go. The job of the transfer RNA is simply to take the guys from one point to another. It's like a taxi. The taxi takes you to the shopping center or to the school. But a taxi driver doesn't have to make sure that you shop at the shopping center or that you indeed go to school even. His job is to drop you outside the gate. What you do after that is your business. So the same way, transfer RNA is a taxi. It goes in the cytoplasm, picks up free amino acids, and specific transfer RNAs pick up specific amino acids. Just like when you go to the taxi rank. There are different taxis for different areas. If you go on the wrong taxi, you're going to go to the wrong area. You see? So you've got to be careful how you do that. So the same way, there is a specific code provided for the amino acid, and that amino acid then will go with the taxi or the transfer RNA to the ribosome, and it will combine, and we'll talk about that also just now. Let's look at the diagrams. That's what messenger RNA, just a stranded structure. rRNA, no real, it's mainly coiled in many areas, and this is what they're talking about, the three clover-shaped type of RNA, transfer RNA. By the way, the transfer RNA, on this side, it picks up the amino acid, and on this side, generally, you have the nitrogenous bases. And it is these nitrogenous bases that will distinguish between different types of transfer RNA and the amino acids that they carry. There. By the way, when we talk about that, we'll talk about that just now in protein synthesis, so I don't want to say too much there. Are we clear with this, guys? Remember, messenger RNA copies the code. Where? In the nucleus. From who? From DNA. It then takes this message to the ribosome, and it attaches itself there. Ribosomal RNA is involved with the production of the ribosome, etc., and attaching the messenger RNA to the ribosome. Transfer RNA simply is there to pick up the amino acids and bring them to the ribosomes. Remember, we said that the ribosomes are the site where proteins are made. In other words, this will be the factory. This is where the proteins are made, manufactured. This is a manufacturing plant, whatever you want to call it. So that's what we are talking about there. Now, when we're looking at this section on protein synthesis, we break it up into two areas. The first area being transcription. Where does it happen? In the nucleus. The second area, or the second process, I shouldn't use the word area here, the first process, transcription, taking place in the nucleus. The second process, translation, taking place in the ribosomes. Let's look at each of this in summary form quickly. What happens during transcription? As DNA replication takes place, messenger RNA forms. How? While the DNA is replicating, when the strands separate, Instead of another DNA forming there, messenger RNA sneaks in before the other strand forms, and it copies the code and it moves away. So it doesn't interfere with replication. Replication will still continue. Okay? This is controlled by enzymes. This messenger RNA then leaves the nucleus and it attaches to the ribosomes in such a way that the base triplets are exposed. They are showing. And the base triplets are known as the codon. They are providing the code. What has happened? Messenger RNA has stolen the code from DNA. And now it reveals that code in the cytoplasm. Why is this necessary? It's necessary because DNA, that nuclear DNA, does not leave the nucleus. So it cannot get the code to the cytoplasm. So the only way this happens is when messenger RNA steals the copy or the code and goes out. The next phase is translation. Now the word, again, English is a simple language in certain cases. Transcription means, the verb it comes from is transcribe. And to transcribe means to take down or rather to copy. So again, from the word transcription, you should know that this is the copying process. Translation, here's a code, we have this code. But if I give you a code and you don't know the combinations, indeed, or you don't know how to, the password for that matter, you can't break the code. So translation means we need something to break this code. If I start talking to you in another language here all of a sudden, you wouldn't understand a word I say. Then I would have to have a translator next to me to do that. So this is what translation means. We're taking this code and we are changing this code to give us something else that we can work with. And that's what's happening here. How does that happen? First of all, the transfer RNA picks up amino acids. Where do they take them? To the ribosomes. And remember I told you earlier, those base triplets, that is known as the anticodon. Now we got two words, the codon, and now we got the anticodon. The codon provides the code, the anticodon breaks the code. But the codon and the anticodon must match. So in other words, if we have here the codon being A, G, C, then the anticodon that comes here should be U, C, G. Remember from our base pairing from last week. But here, remember, look at the special case here. We said at, cute girl. In RNA, this doesn't apply because there's no at, there's no T. So whenever there's A, it will combine with a U. It will not be replaced by a U, but it will be combined with a U. Just keep that in mind. The codon, the amino acids then come next to each other. They join. 2 will be dipeptide, 3 will be tripeptide, many will be polypeptide. And when this polypeptide gets to 50 and more, we have a protein. All of these reactions are controlled by enzymes. And when 2 amino acids join or combine, water is released. This is known as dehydration synthesis. And again, a picture says more. There's your story. DNA molecule is unwinding. Same story as replication, so that's what's happening. As it unwinds, the messenger RNA, the messenger RNA starts forming here with that. Here's the one with the blue backbone. See here, with the C came G, with the T came A. The T was in DNA. We do not say now when there's a T we must put U. No, T always combines with A and this continues. A, now here, A normally combines with T, but in RNA there's no T, so that is replaced with a U, and so it goes. That's the messenger RNA forming. What does this messenger RNA do? It then moves out of the nucleus. There we go, it's on the left-hand side there, sorry. They move out there and they go out here in this area here, and we have the messenger RNA formed. When it goes out of the nucleus and it aligns on the ribosomes. With that, guys, I think you deserve a break.
Sure, that was amazing. I say carry on. Hey, no, okay. Guys, jump up and down if you're being lazy and you're lying on the couch. I think it's time to get some energy. I think I might even do that during the break. Post your questions on the Facebook page, and we'll see you now shortly after the break. So now, Aslam, this question is from Kalobe, and she says, I don't understand this one. What is the difference between DNA transcription and the DNA translation?
Okay, that's Kolobe.
Yes.
Kolobe, first of all, we don't talk about DNA transcription and DNA translation. We talk of transcription and translation. It's actually involving RNA and not DNA. Although DNA is involved in the process, but the actual transcribing is done by RNA. And again, we go back to our diagram to highlight that explanation. The first part is transcription, and that's occurring in the nucleus, as you can see. And transcription means to transcribe, to copy. So transcription is the process whereby messenger RNA copies the code from DNA. In that one sentence, that's what transcription is all about. Obviously, enzymes are involved in this process, and obviously you have to explain in a longer question that DNA unwinds, and as the DNA unwinds, the nucleotides of messenger RNA attach to one of the two templates, and it then breaks away and goes away out of the nucleus into the cytoplasm. That is transcription, and that's what's happening there. If we go further down, then what happens is that the messenger RNA attaches to the ribosome, and we have what we call transcription taking place. Just to backtrack on the question as well, just to understand what's happening. DNA provides the code. The code then is transcribed. Then it was translated to form a protein, and a protein forms the tray. The tray being not a tray that you're carrying around, but the characteristic, blue eyes, green eyes, etc., etc., etc. So that's what's happening there. Now, transcription, on the other hand, just need to extend this page a bit, because we've got one more question, I think. Transcription, if we can get some writing done, it would be good. Let's write here, transcription. Transcription. Here now, we're at the ribosome, and messenger RNA is arranged there, base triplets showing, exposed. So if that's A, G, C, 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 G, A, U, G. That is what's happening to the messenger RNA. And transfer RNA, in the meanwhile, it's there, and it also has bases. If it has U, C, G, it's going to attach to this one here, to this base triplet. And with it comes an amino acid. And with another transfer RNA, another amino acid. These two amino acids, transfer RNA just brings them together. They then bond. They form a peptide link. Water is lost. We said dehydration synthesis taking place. And so on along the chain. So what is happening? We had a code. This code is being translated to another language, if you want to call it that. And that language is called formation of a protein. And that is translation. So transcription is the actual, it takes place first of all in the nucleus, copying the code. Translation is using that code to manufacture a protein. The other question was?
Okay, and the other question was from Letlonolo, and he asks, why are there no hydrogen bonds present in RNA?
Great, guys, I'm surprised. Did anybody answer the question for you?
There has actually, it's Wilhelmine has answered, and so has, no, it's just Wilhelmine. And guys, I love that. I love that they are
What's their
helping.
answer?
Okay, so his answer was because it is single-stranded.
There you go. Why do you need me?
Exactly.
Just let them
No, well,
answer
we
the
do
question.
need you. We do need you. Don't go anywhere.
There you go. Great. What was the person of Wilhelmine? Eh?
Yes.
Thank you, Wilhelmine. That's an excellent answer. And that is the difference between DNA and RNA. What is the main difference? One of the main differences is that DNA is double-stranded. It's made up of two strands, whereas RNA is single-stranded. So if you had double strands, you needed something to bond the two strands together. When the molecule is single-stranded, there is no need for a hydrogen bond. I'm happy with that answer. That's a great answer. That would have earned you, Wilhelmine, full marks in an exam if that was the question.
Fantastic.
I think that was the last question, eh?
That is the last question.
Good. Okay, we move on, and I'm glad we came to this stage of mutation, and you saw earlier we had a mutation. Mute, you couldn't hear us on the other side. So what happened? Something went wrong. Mutation. Something goes wrong. So a mutation is when something changes in this process that we are talking about, whether it's in DNA replication, whether it's in protein synthesis, or whatever we're talking about, we can have a mutation. Something can go wrong with the code. So first of all, it can happen in a DNA, or it can happen in a chromosome of an organism. If it happens in the chromosome, it's called a chromosomal mutation or a chromosomal aberration. Big word. If it happens in a DNA, it's called a gene mutation. Gene mutation. So there we have the definitions there. Gene mutation, a change in the DNA or the gene. Chromosomal, a change in a larger section of the chromosome itself. And when we talk about later meiosis, when we say when there's one more chromosome or there's one less chromosome, that is also a chromosomal mutation because it involves a whole chromosome in that case. What causes this, you ask? Many. Sometimes this is spontaneous. Spontaneous meaning automatic. Just something goes wrong with the code. Just like what happened earlier. Nobody pulled anything out. Nobody pulled a plug out. Something just went wrong. It's spontaneous. Sometimes it could be because of environmental factors. And in the technological world that we are living in, you'll read a lot about X-rays, radiation from cell phones and the like. And we are using a lot of equipment. We are using a lot of technology. And come from the technology, there are signals that are being emitted. And these signals can disrupt the way things are happening inside our bodies. We need to think about that as well. And from that, we have UV light, X-rays going. People that work with X-rays, people that go for X-rays, you'll notice when they go there, they have to wear specific clothing, etc., 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 so that they do not get too exposed to those, the radiation and the X-rays that come out there. Chemicals can also cause problems. Chemicals like benzene, formaldehyde, carbon tetrachloride. The chemicals in cigarette smoke are also seen to be causative, or what we call a mutagen. Something that causes a mutation is called a mutagen. Just like something that causes cancer is called a carcinogen. So the same way, and cancer, by the way, is a mutation. The cells don't know that they have to stop dividing. They are uncontrollably dividing, and they form a tumor, and they spread to other areas, and the whole body can get affected by this because they press against other organs. If you have a tumor in the brain, for example, if it spreads or if it presses against a part of your brain that is dealing with eyesight, then you may lose your eyesight, etc., etc. So all of these things cause mutations. The two types of mutation that are related to protein synthesis we're going to discuss. But before we go there, I just want to look at three other types of mutation. And these are the more general. I'll write it here. Lethal, neutral, and fixed. And these three we look at here, we look at in meiosis, we look at in evolution, we look at it all the time. Lethal, by now you should know, lethal means it's going to cause death of the organism. It is so dangerous, it's so harmful that the organism cannot live because of it. So it's going to kill the organism. Neutral, on the other hand, if we say somebody is neutral, that means they are not on this side, neither on the other side. If your car is put in neutral, it's not moving forward, nor is it moving backwards, unless, of course, you are leaving it in neutral and have it facing downwards or backwards downwards on a hill. Then obviously it's still going to move. But if you leave it on a flat surface, it should not move. However, there are certain places on the earth, in certain areas because of certain forces. They leave the car in neutral. I've seen it, I've sat in a car which moved uphill in neutral. All right? But generally you get the idea that in neutral the car is not going to move. So neutral means there's no effect. It has no effect. And fixed generally is a beneficial mutation. It's beneficial to such an extent that it gives the organism that developed because of the mutation an advantage over all the others. So that organism survives, and the ones without the mutation, they die because they do not have the special advantage. And because of that, the rest of that species will continue with that mutation. Therefore, we say it's fixed. It remains with the species. However, in protein synthesis, whenever they link mutation with protein synthesis, then you must look at these two. And they are a point mutation. And a point mutation means, as the word says, point. A point mutation means that a particular nitrogenous base is replaced by another one. For example, in this example here, G is replaced by A. There was some mistake took place, and G was replaced by A. But look at the consequence of that. That was GGC. This is AGC. Now, because of this code, obviously the transfer RNA that comes here would have to be different. Here, the transfer RNA that will come here will have C, C, G. Now, we have to have U, C, G. Originally, that transfer RNA, CCG, was coding, for example, for glycine. And this one now, UCG, codes for serine. So the type of amino acid on this chain changes, but only one amino acid changes in this chain. However, it will still be a different protein from the one above. And because of this, the protein will have different functions and structure, etc. But it's only on one point, remember that. Now, some people say, yeah, if it's on one point, then it can't be very bad or very good. But look at this case here now. This is a classic example of a point mutation. Here we have DNA, RNA. We're having this one here, having that particular code. And because the A is replaced with a U there, we have valine forming there. And because of this, we have abnormal hemoglobin forming. And what does this do? It changes the shape of the blood cells, red blood cells. The red blood cells, instead of being biconcave, this way, they become sickle shaped, like a sickle. And because of this change in shape, the blood cells do not flow properly, and they cause clotting and other issues among the patients that have sickle cell anemia. Now, for us, sickle cell anemia is harmful. But in certain areas, especially in Africa, sickle cell anemia is seen to be an advantage. The people that have sickle cell anemia, for example, they do not suffer from malaria. In areas where malaria is prevalent, then the people that have sickle cell anemia, this seems to be an adaptation for them. It's a suitability for them. They survive because the malaria parasite cannot attach too nicely to the sickle cell shaped blood cells, and therefore they do not suffer the consequences. And then that's one letter. It was just one letter that changed, not a whole sentence, just one letter. All right? Keep that in mind. The next type of mutation is known as a frame shift. Now, again, English, guys. That one was a point, so it was 1 point. Frame shift, frame shift. The whole frame shifts. How does this happen? This happens if, for example, in this example, we take away this U here. So that means if this thing was reading AUG, AAG, we took out the U, it was UUU, it became UUG. So that's the next code. And so on, that everything after this steps in one way. And when they step in one way, the whole sentence thereafter is different. And that causes, again, a change in the sequence of every amino acid thereafter. And the whole protein will change. And in some cases, this protein may not be functional even. It could be functional, could not be functional as well. Another way that we can do this, instead of deletion, as this one was, if we insert something here, if we insert an A here, then obviously the next word if we want to call it a word, will be AGG. And then CGC, the same effect. The whole frame thereafter changes. So when we insert or delete a nitrogenous base, then the whole frame changes thereafter. When we replace one nitrogenous base with another one, then it is only that point that is affected. So that's the difference between a point and a frame shift mutation. All right, we go straight to the questions then. Let's look at the first one. I'm only going to look at the correct biological terms. Nitrogenous base, which is found in RNA, but not DNA. That one is easy. It's uracil. A triplet of 3 bases found on an mRNA, what we call the collective. We call it the codon because it provides the code. The single strand of DNA that contains the instruction for making a protein, the one that shows the code, we call it the template. The template or the mold. The type of RNA that picks up amino acids in a cytoplasm and brings them, that's easy. Picks up, the word picks up tells you something about what I told you earlier, the transfer RNA. And factors that cause mutations in DNA and chromosomes are called mutagens. The synthesis of mRNA from DNA, formation of mRNA from DNA, this is known as transcription. Transcription. Tabulate the differences between DNA and RNA. We taught you last week how to draw the table. We discussed quickly the differences. One is single-strand RNA, double-strand DNA. Uracil found in RNA, thymine found in DNA. No hydrogen bonds in RNA, hydrogen bonds found there. Helix shape in DNA, not helical in RNA. So those are some of the differences between DNA and RNA. And with that, guys, I think you have a mouthful. Look at the page with the notes. There are some other questions which you can look at, are valuable. With that, I give over to Indy.
Thank you so much, Awesome. Okay, guys, don't forget that this is proudly sponsored by Macmillan. Thank you so much, guys, for making this show possible. You guys rock. Also, we've just posted onto the Facebook page. Post your random acts of kindness on our Facebook page, and you could win fantastic prizes. Watch Learn Extra Live Monday to Thursday to see if you are a winner. So that's what you do, guys. Do something nice. Post onto our Facebook page, photo or video, and you could win some great stuff. Thank you so much, Grade 12s, for such an awesome life science lesson. It's been amazing. See you same time, same place next week. Goodbye.